Morimoto is worldwide recognized for his studies of molecular chaperones and the regulation of stress response in physiological and environmental stresses. He studies how these stresses could lead to neurodegeneration. Morimoto's scientific activity led to more than 200 well-cited manuscripts, among them a review that was first to describe the term proteostasis. He won many awards, and since 2011 is a fellow of the American Academy of Arts and Sciences. Just a reminder, as in previous webinars, you will be able to ask questions right after the presentation, but you first need to type the questions on the webinar control panel. And just to let you know that the next webinar will be by Alfred Goldberg from Harvard University on February 4th, 2016. What's going on?
We switch now to myself?
Yeah, you are.
Okay. Good morning, everyone from Chicago. It's Rick Morimoto. Happy New Year. Although I'm sure all of you are familiar with the origins of proteostasis, I thought I would begin 2016 and my seminar by going back to that original paper where Jeff Kelly, Andy Dillon, Bill Balch, and myself came together to establish this important new concept. Let's see, let's see, control my slides. Let's go. Okay. The proteostasis network is more than just the concept of how polypeptides go from highest free energy states, the unfolded, to the folded protein. We recognize that an understanding of proteostasis really began with gene regulation because that influences the concentration of mRNAs and, of course, consequently, the level of translation of different proteins or solutes that influence solubility within any particular compartment. You'll notice that in these slides, there will be two different colors. A red ball, which corresponds to areas where we feel one can identify small molecules that we termed proteostasis regulators, and the green squares, which correspond to pharmacological chaperones, the best example being tafamidis, which Jeff Kelly discovered to stabilize transthyretin from misfolding. We recognized from the outset that the complexity was driven by unimolecular misfolding off pathway to non-functional proteins, and that these non-functional proteins would then have to be cleared either by the proteasome or autophagy, and the recognition of off-pathway to aggregates. These aggregates, of course, can take many different forms into the oligomeric state, into the protofibrillar, and eventually the fibrillar state, or just mixed heterogeneous aggregates. The last part, of course, has to do with the ability to stabilize folds, in this case, most certainly, and demonstrated by tafamidis for transthyretin, but also the importance of transport. So you can already see that proteostasis as a concept expanded beyond traditional concepts of quality control and really embraced the multitude of steps that were essential to guide stability of the proteome. Our ideas are embedded deeply in physical biochemistry. So this is an energy landscape. And in blue, you can see that the unfolded polypeptide from the highest free energy state goes to the lowest free energy state. The blue arrows correspond to places where molecular chaperones indicated in networks below then guide the intermediates towards the lowest free energy states. In red is the formation of off-pathway aggregates, where it's already clear that the ability of chaperone systems, such as the disaggregases, of the A and B class J chaperones, or the HSP 1010, shifting the 70 machine towards disaggregation, now convert highly ordered aggregates into monomeric species. How this is regulated is tied into signaling. This is an example coming mostly from Andy Dillon's work and my laboratory and others, where the role of insulin signaling, age 1 DAF2, requiring both HSF and DAF16 to achieve longevity. The important concept here was that lifespan itself required a stable, healthy proteome. And of course, the critical role of other transcription factors such as FA4 and SKIN1. SKIN1, of course, is NRF2, tied in as antioxidant stress response, linked, of course, to mitochondrial function and consequences. Another way to think about this in a much more narrow is then the transition of proteins from the unfolded to the native state and off-pathway intermediates that form these oligomers and aggregates. Using a more traditional view of a landscape diagram, you start with the highest free energy state, the nearly infinite number of possible conformers, the intramolecular context in light blue going towards the native state, where you still have these intermediates that require ATP, energy, and chaperones to drive them to the native state. And in the darker blue is the oligomers and amyloid fibrils and amorphous aggregates. And what's important to recognize is that for every polypeptide, depending on its environment and its solubility, that proteins, and its mutational state, by the way, that a protein can shift from intramolecular to intermolecular contacts. What determines this, of course, are the molecular chaperones, and in humans there's 341 of them in 9 gene families, and the major clearance mechanisms that you know well of autophagy and the proteasome that prevent off-pathway and guide and direct towards functionality. The challenge in biology is the number of polymorphisms that we encode both in the protein coding regions, but as well polymorphisms that affect the expression, increase the expression, or alter the kind of protein by affecting splicing. Biosynthetic errors often we don't think about, but it's critical because they, every process of the many dozens of steps from gene expression to protein folding involve steps where each one has a error-prone frequency in the range of 10 to the minus 4th to 10 to the minus 5th. So if one thinks about polymorphisms and biosynthetic errors, that itself adds a significant load to the concept of signal, noise, and quality control, affecting protein damage and aging. Proteostasis, then, is a concept you can understand, is balance. How do you achieve function in the face of dysfunction? With stress and aging, the balance is tipped towards dysfunction, and that's what I think many of us are intrigued to understand. When does this occur? How does this occur? And can one reset the system? In more detail, this is just a cartoon that gives a deeper sense of within any single cell the complexity. So up in the left-hand corner here is the beginning where off the mRNA bound to the ribosome, you have nascent chain synthesis interacting in eukaryotic cells with alpha and beta NAC, and it's been well demonstrated from a number of laboratories the importance of alpha and beta NAC, very much like trigger factor in prokaryotes, to welcome the nascent chain to allow initial steps of folding. You then get the interaction with a set of molecular chaperones. If folding occurs properly, you end up with a protein indicated in this case in green, which can then interact with subsequent chaperone networks, usually the 70 and 90 system, to generate a functional complex. If, however, the protein's misfolded or the environment is altered, it can interact with the small heat shock proteins. It can then interact with the disaggregates HSP110, HSP70, HSP40, which then reverses the process, or it can go forward either to the autophagic machinery, as indicated down below, or it can shift over to the triage machinery, where interaction, for example, with CHIP and other E3 ligases then directs the protein towards the proteasome. It's at this point of the proteasome that one engages both cytoplasmic events and events in the endoplasmic reticulum, because through ERAD you get a redirection of proteins that are improperly folded out of the ER back into the cytoplasm, where you get competition with cytoplasmic events. Likewise, the mitochondria has its own set of chaperones as a symbiote, and there's a major role, of course, for chaperones in importing polypeptides into the mitochondria and interaction through mitophagy with the lysosome, where you get an engagement of competition with the same reactions. One of the important aspects then of proteostasis is that instead of looking at individual steps or processes, which has been the tendency, is the recognition that an understanding of proteostasis is going to be how do chaperones shared in all these different processes engage and determine stoichiometry and balance, or in other cases, competition. That sequestration of chaperones in one reaction is going to have profound consequences throughout. This helped us sort of think about a cartoon shown here, where the proteostasis network, which actually is well beyond just molecular chaperones. It includes the autophagic machine, the ubiquitin proteasome. It involves transport mechanisms. So you're talking about some 1,500 to 2,000 genes are critical, and in each cell provide the network that allows a protein, despite its polymorphisms and mutations, to fold and achieve function. We feel that a lot of this is going to be highly cell or tissue specific. On the right-hand side in red is when the proteostasis network is challenged, either by aging or disease-associated mutations. What this sets up is competition in one case, or a decline in the case of aging, where the system now starts getting shut down during senescence, leading to an alteration of the proteostasis network, leading to an inability of proteins with polymorphisms to fold functionally, leading to cell-specific dysfunction. This, of course, brings forth the nearly 600 different genes, pathways, and systems that are highly dependent on proteostasis and fail when you have an abundance of misfolded proteins. While much of the emphasis has been on the nervous system and on proteins such as A-beta, tau, alpha-synuclein, on, one has to keep in mind that the same concepts extend to cystic fibrosis, to beta-2 microglobulin, to prolactin and insulin, and to diseases such as COPD, emphysema, and arthritis. So the common feature then of proteostasis and its importance, I think, is perhaps highlighted by aging in the human brain. And shown here is just a cartoon to remind us of the histopathology of the normal human brain and in age-associated Alzheimer's disease. And to the right, a series of images indicating the level of plaques and aggregates that appear, whether due to misprocessing and overexpression of A-beta or tau or the PRP-SC form or alpha-synuclein. But to make the point that it's not just neurodegenerative disease, but diseases such as COPD, respiratory diseases are associated with rather substantial stress to biological systems and impairment of proteostasis. And of course, the impact on the cardiomyocyte as a function of aging, leading to increased autophagic cell death, hypertrophy, and damage to the cellular system. In each of these cases, there are abundant examples that the risk factor is the transition of normal proteins towards toxic states, either the loss of the protein or the fact that aggregates itself then cause problems to cellular function. One, of course, doesn't need to be reminded that many diseases indicated here, cancer, cardiovascular, flu, and Alzheimer's disease, are all age-associated and exhibit complex morbidities in the elderly population. This, of course, leads one to recognize that as a function of aging, there's a collapse in proteostasis, and the consequence of that is a loss of cell and tissue function. Early onset of proteotoxicity, of course, is the consequence of environmental risk and genetic mutations. The goal is really to push it to the right. Could one enhance the cellular proteostasis machinery? Could we identify the genetic risk factors, the imbalance early, the proteotoxicity that indicates who in the population could be treated by proteostasis regulators and to suppress such diseases? So early on, studies in our laboratory that were done by Tali Gedalovitz and Anat Ben-Zvi demonstrated the importance of the stability of the proteome, demonstrating that under normal circumstances, a genome that encodes polymorphisms indicated as these red spots in the genome result in folding variants. These could be called temperature-sensitive mutant proteins. But that the normal capacity of the quality control machinery allows these folding variants to fold properly unless the system is challenged. And in this case, the introduction of a single allele with a polyglutamine expansion or mutations in SOD1, for example, now create a competitive environment. The growth of this aggregate then interferes with the ability of these mild folding variants to fold properly because the quality control machinery is overwhelmed. The chaperones, instead of being available for these folding variants, are now functionally sequestered by the aggregates, leading to both gain of function toxicity and loss of function when these mild folding variants do not fold. We put forth early on that this idea of chaperone competition contributes, we think, then to as a common feature of disease mechanism. Much of our work and much of what I'll describe to you is based on our work in C. elegans, and I think many of you are familiar that we've made many dozens of models using C. elegans. An example shown here, a short polyglutamine stretch Q19, a long polyglutamine stretch Q82, generates very dramatic phenotypes. Either in one case, below threshold, as indicated with Huntington's disease, you get no phenotype. You cross the threshold as shown below in the graph. As you increase the length of the polyglutamine alone, as you cross over 33 to 35 glutamine residues, you now get this very striking age-dependent aggregation. Another 5 residues of polyglutamine, and it becomes a very dramatic both aggregation and toxicity. You can see how the age effect is quite striking, that the expression of a protein just at the threshold of Q35 does not generate any aggregate species. The protein is soluble, it's a monomer, but in an age-dependent manner, you now get the appearance of these. And you can see back in the graph below that as the animals age, you now see with a short polyglutamine Q29, no toxicity, Q33, Q35, no toxicity during development, but as soon as the animal becomes an adult, you generate toxicity, whereas a longer polyglutamine, they're toxic at an even earlier stage. This led Jim Morley to make an important observation that perhaps the proteostasis network itself was a key component of lifespan pathways, and immediately looked at the insulin signaling pathway regulated by DAF2H1 and demonstrated, shown here with animals expressing the highly toxic Q82. So this is an embryo. Here's the adult animal with the aggregates, that if one simply introduces the mutation that makes these animals long-lived, you now suppress aggregates. Moreover, he demonstrated that this was absolutely dependent on 2 transcription factors, DAF16 or FOXO3 and heat shock factor 1. And together they provided stress resistance and regulated protein aggregation in providing lifespan. Our studies in C. elegans then have, I think, provided for us a number of insights, and perhaps one that we find most intriguing is that instead of thinking about the animal as just a collection of cells, the recognition that the cells and tissues are communicating their proteostasis status between the tissues to determine organismal health. And this is really demonstrated in this cartoon here, where even though all cells and tissues express heat shock factor and molecular chaperones, environmental perception of heat shock is really sensed by the thermal sensory neuron AFD, and that transmits a signal to the other tissues. Likewise, other tissues such as the muscle sense folding and misfolding, for example, of a mutant myosin, and other tissues then compensate and activate chaperone systems that are protective. So I'm just going to give you a few examples. The first is by Veena Prahlad, who discovered that the heat shock response in a metazoan animal is not regulated cell autonomously, but rather cell non-autonomously by the AFD neuron. So if one genetically ablates this neuron, there's no heat shock response in any other tissue, even though the heat shock factor is present and can be activated by other forms of stress. Most intriguingly, if you remove this signal, then the entire system goes cell non-autonomous, and each tissue then decides on its own how to activate the heat shock response. Veena most recently, earlier last year, demonstrated independently in her new lab at Iowa that this is regulated by serotonin. So this, of course, being a conserved neurotransmitter pathway, I think provides a great insight into how communication occurs between neurons and the non-neuronal somatic tissues. Another observation by Patricia Van Oosten Hall was that if one alters the levels of chaperones in tissues, to our surprise, other tissues recognize that imbalance. So, for example, if one reduces the amount of HSP90 either down or up genetically in a specific tissue, body wall muscle cells, the neurons and the intestinal cells actually recognize this and activate a compensatory response. If there's too little chaperone, they activate more. If there's too much chaperone, they block induction of the heat shock response. The consequence of that is an organismal net benefit and coordinated response. Leads to the idea of ascending tissue and receiving tissue in transcellular chaperone signaling. A third example is a genetic discovery where we take animals expressing a misfolded protein in the postsynaptic muscle tissue and then ask what tissues communicate information to restore misfolding in the muscle. That led to the discovery that it's the balance of the cholinergic signaling. In other words, the balance of acetylcholine and GABA as the inhibitory neurotransmitter determine the level of calcium flux going through the voltage-gated calcium channel, release of calcium from ryanodine receptor, activation of HSF by the calcium-dependent kinases and calmodulin, leading to the enhanced expression of chaperones and restoring folding stability in the postsynaptic tissue. And most recently, Janine Kirstein, when she was a postdoc, did a very nice study demonstrating that redox was also regulated not only between different compartments, so between the cytoplasm and the ER, could recognize an imbalance, but transcellular stress, that if one imbalance redox in one tissue, other tissues recognized this and activated a protective mechanism. Together with Ulrich Hartl, we then launched a proteomic study, and just as a brief summary, Ulrich's laboratory looked deeply at the proteomic analysis in wild-type young, aged animals and in long-lived animals. And the striking difference in long-lived animals is not that there's no aggregates. We still get age-dependent aggregates. There's a decreased translation that occurs in the long-lived animals, and there's one other very important feature. There was about a 30-fold induction of small heat shock proteins that associated with the aggregates, and we think that what this does is it restores the imbalance due to misfolding and aggregation. And this just shows you some examples of the data that one sees very early, looking at the cytosolic ribosomes, the mitochondrial ribosomes, the 20S proteasome core, the 19S cap, that there's a decline in the ribosome subunits, and an increase in proteasome. And you'll notice that most of the change actually occurs very early in adult life. Moreover, if you look at chaperones quantitatively by mass spec, you see that most traditional chaperones, the HSP70s in different compartments, the mitochondrial, the ER, the cytoplasm, and HSP90 really don't change in any significant way, but it's the small heat shock proteins that go up quite dramatically in response to aging. What this reveals to us is that there is a loss of proteome balance, but it's really an alteration in protein stoichiometry, that there are changes in protein abundance leading to extensive protein aggregation during aging. And we think what's going to be critical is that there's a sequestration of surplus proteins in chaperone-associated aggregates by these small heat shock proteins that we think is going to be critical. This, of course, has allowed us to return to an interesting question initiated by Anat Ben-Zvi when she was in the laboratory, using the temperature-sensitive mutant proteins to ask the simple question, when does misfolding occur? If one looked at the literature in aging biology, there was a tendency to think about changes in mid to late aging, whether it's a mouse, a fly, or C. elegans. So it was a surprise to us when we looked at various temperature-sensitive mutant proteins kept at the permissive temperature of 15 degrees, that, for example, wild-type paramycin, by immunostaining, the myofilaments are fine. You can see the animals move fine. There's no disruption. By day 12, or about 2 weeks, you start to see some myofilament structures that are altered. But in animals harboring a single missense mutation in the helical region of paramycin, I think you can all see that the paramycin forms aggregates as early as 3 days and dramatically by 5 days. We could actually quantify this by the disruption of the 95 body wall muscle cells, and you can see that 50% of the muscle cells were disrupted between days 3 and 4. In other words, early in adulthood. This was not a late phenotype. Anat went on to look at many, many other proteins. It's just showing you an example of other proteins, RAS, acetylcholine in neurons, GAS1, a mitochondrial matrix protein in neurons, or perlecan, a muscle protein. And in the top is always the phenotype for the TS mutant protein. The bottom is the wild type. And you can see in every case, the metastable TS mutant protein misfolds and loses function early in adulthood. This then led us to ask the question, what's happening? Anat had demonstrated that there was a decline in the heat shock response. John Labadia then asked the question exactly when this occurs. And you notice here that we're looking at adult animals that are 4 hours old, 8, 12, 16, and 20 hours. We look at induction of the heat shock response by qPCR. You see that 4 and 8 hour adult animals have a full robust heat shock response, but that it declines dramatically by 60 to 80% within 4 hours. And this is reflected at the protein level. You can see a Western blot that, in fact, the inability to activate heat shock genes is reflected by a reduced level of protein. Where this is most dramatic is if you ask what's the physiological consequence. So animals that are 4 or 8 hours old exposed to a 5-hour heat shock at 35 degrees are 90% resistant, whereas in these animals that don't have a heat shock response, have declined levels of chaperones, you see that 90% of the animals are now dead. In other words, the inability to have a robust heat shock response in this 4-hour period really is life or death. And I think this is reflected here in this movie, where if one takes animals at day 1 or day 2, shown in this cartoon, this movie, you can see that the animals at day 1 exposed to heat shock are stress resistant. Day 2, they're actually dead. Now, what's dramatic here is that this timing of a decline of heat shock that you see corresponds perfectly and inversely to reproductive maturity, because it's between day 8 and day 12 that the animals now become reproductively mature, as demonstrated by egg-laying. And we'll get back to this because this turns out to be key in understanding what we think is the beginning of aging. So we did a whole series of molecular biology. What's the basis of this? So shown here is that the level of HSF1 mRNA doesn't change at all. The level of HSF DNA binding in vitro by what's called gel mobility shift assay in control heat shock day 1, control heat shock day 2 is completely unaffected, so there's no problem with the ability for HSF to form trimers. We look at the levels of HSF1 expressed in a single copy HSF1 GFP fusion protein, and you see on day 1 it's in the nucleus, day 2 it's in the nucleus. Upon heat shock they form stress granules. Where we find a dramatic difference is when we look in vivo at the chromatin and look for occupancy of HSF. You see a dramatic reduction in HSF binding between day 1 and day 2, a dramatic reduction in RNA polymerase binding by CHIP chromatin immunoprecipitation analysis. This corresponds to a significant reduction in DNase hypersensitivity at the HSP70 promoter between day 1 and day 2. And you can also see that when we looked at all the different chromatin marks, acetylation and methylation, we saw only one change, and that is a dramatic increase in H3K27 trimethyl marks at the stress gene loci, shown here representatively for HSP70 and HSP16. Now, this repressive mark is also known as polyQ, and this is a very important mark which normally in development is used as part of how one gets differentiation. You shut down entire pathways. So the fact that this H3K27 is being utilized in adulthood to shut down stress gene loci is obviously intriguing. Now, the balance of methylation requires, of course, the demethylases, and in C. elegans they encode 4. Only one of these declines between day 1 and day 2. The other 3 do not change. If anything, they go up slightly. And we notice that at the RNA level, Jumanji 3.1, the specific demethylase, actually starts declining before we see repression. So it's absolutely consistent with these chromatin repressive marks. We see a decline between 4 and 8 hours, and of course this decline corresponds then to an increase in H3K27 marks. Now, if you look at animals that are deficient in this demethylase, you find that the heat shock response, in fact, is reduced on day 1 and is no longer further reduced on day 2 because you already have maximal repression. So we decided to ask, could we retune the system? Can one reset proteostasis? So what you're looking at is the induction of HSP70 RNA in day 1 animals in the demethylase mutant, and when we now overexpress the demethylase. So when we overexpress the demethylase, of course, you're no longer getting H3K27 marks. You now get you're back to the normal level of HSP70. What's critical is to look at day 2 and day 3. This is the normal decline between day 1 and day 2, and you now see that by just overexpressing the demethylase, we now restore dramatically the induction of HSP70. Moreover, we restore thermal tolerance. So you see this very striking reduction in thermal tolerance, as I pointed out earlier. When we now overexpress the demethylase, there's no longer repression of heat shock loci, and the animals are now stress resistant. What's important here, it's not just the heat shock response that's restored. There's also a repression in the unfolded protein response in the ER, the unfolded protein response in the mitochondria, and the antioxidant stress response. When you then overexpress this demethylase, you restore resistance to DTT, restore resistance to ethidium bromide, and restore resistance to paraquat. So we think that this is not just about heat shock, but it's about all stress loci that are repressed at that same moment. Then comes the final question of what's the signal. And so I pointed out earlier to you that we knew that there was an inverse relationship between the loss of the heat shock response and reproductive maturity, which of course is regulated by the germ-line and the various reproductive organs. So we looked at genetic mutations that are germ-line stem cell deficient. 2 different mutations, GLIP1, which is Notch, or GLIP4. And what you can see here first is that in the open is the wild type. You see the decline in the heat shock response. But if we now look at the GLP-1 or GLP-4, day 1, day 2, there's no repression. Day 3, there's no repression. Day 4, there's no repression. In other words, if there's no signal coming from the germline stem cells, the stress responses are completely protected and persist. Moreover, at the molecular level, we can show that we get a complete restoration of RNA polymerase II binding to the heat shock gene promoters in the GLP-1 mutations, consistent with a molecular restoration. Moreover, the decline that we see in Jumanji 3.1 demethylase in wild-type animals between day 1 and day 2, where you can see it is low levels, is now essentially fully restored in these animals without any germline signaling. We know that this signal is specific because animals deficient in oocytes genetically, or sperm, or gonads, still have the block in the heat shock response, arguing that it's a highly specific signal coming from the germline stem cells to the somatic tissue that then inhibits the heat shock response. So this is for those of you who are less familiar. This is a cartoon of C. elegans. These are the germline stem cells that then generate the oocytes, the sperm, and then are fertilized in the spermatheca to generate the next generation of progeny. So a cartoon that sort of summarizes this thinking is really that Jumanjis play a critical role to clean and maintain the chromatin, allowing heat shock factor to bind, and then that there's a trigger that's coming from the germline tissue. We don't know what that is yet, and it blocks the ability of HSF to bind. And what's intriguing about this is that it doesn't happen late in aging. It's happening at a very precise little window in time in reproductive biology, suggesting that maybe this is one of the first events that initiates the loss of stress resilience and aging. This, of course, I think brings up many questions. Perhaps the most interesting of these is what happens in the human tissue. So I'm just going to summarize a few bits of data that many of you might be familiar with. Mark Bremy then, together with Mark Vidal's laboratory and colleagues at Proteostasis Therapeutics, looked at the frontal cortex of the human brain. We're looking only at human molecular chaperone RNA expression. Red means it's expressed at a high level. Green means it's repressed. You're only looking at the 332 chaperone genes in humans from age 20 through 99. And I think what you can all see is that there's a dramatic reduction in the expression of about 100 molecular chaperones that corresponds mostly to the ATP-dependent energy-requiring chaperones. Moreover, if we then take independent data sets from Alzheimer's disease, we find that the genes that go up in aging overlap significantly with the genes that go up in Alzheimer's disease. Moreover, the genes that decline in aging overlap significantly with a separate data set of genes that go down in Alzheimer's disease, suggesting that there may be a real link between age-dependent changes in the chaperone and disease-associated changes. Just to give you a sense of where we're thinking and going on this, this is data from the GTEx, where this is from 160 different humans, post-mortem tissues, 30 different tissues. And what you can see here is the expression of the human molecular chaperones in different tissues. What's dramatic is all the brain tissues look almost identical. They're not exactly, but strikingly different than all other tissues of the human, indicated over here, arguing that the brain always looks fundamentally different in which chaperones and which components of the proteostasis network are expressed. We do find a very close relationship among lineages. Muscle tissues map together more closely. Adipose tissues and epithelial tissues map more closely. Not all brain tissues, the pituitary, the cerebrum, and cerebellum, are similar but very distinct, for example, from the caudate, from the amygdala, and the putamen. the concept of how to think about this, it was very much brought forth by studies that Prajwal Siriam, an MD PhD student in my lab, did together with Michele Vendruscolo and Chris Dobson, creating this idea of supersaturation, where we put together the use of this algorithm, the Z aggregator score, which looks at every protein, to actually pose a simple question. If you look at aggregates, are the proteins in aggregates intrinsically aggregation prone? In other words, do they exhibit saturation? Will they naturally fall out of solution? And what was to our surprise was that if you looked then at archival data for Alzheimer's disease, Parkinson's disease, tauopathies, and looked at the other proteins that are in these aggregates and simply used the Ziggregator algorithm, we discovered that they had extremely high supersaturation predictions, arguing that they were in aggregates not by accident, but because intrinsically they were highly aggregation prone. So what this has led us to is really this idea that the network that I've described to you at the organismal level is really about tissue communication, as I pointed out at the beginning. And the tissues have the ability to recognize the proteostasis capability of other tissues, both beneficially and to inhibit the system. During aging, at least in C. elegans, we know that the network is genetically regulated by a signal, a specific signal coming from the germ-line stem cells at the point of reproductive maturity that genetically shuts down by epigenetic repressive marks multiple stress gene loci. But intriguingly, we can reset the system. We can do it genetically, and we would hope that we can also do this by using small molecules to reset the system, to restore stress resilience and enhance longevity. So I think in closing, our studies and the contributions from many laboratories and all of you, I think, have provided insights into how proteostasis is essential for cellular health to prevent stress and age-associated proteotoxicity, and that our ability to understand this, in a sense to manipulate this, may provide ways to think about enhancing tissue robustness and organismal lifespan. So many people in my laboratory contributed to these studies. I have wonderful collaborators. I've mentioned the names of many key scientists who were former members of my laboratory who contributed to the discoveries. A lot of collaborators around the world at many different institutions and funding. And with that, thank you very much.
You can now ask questions by typing questions on the webinar screen. Let's see. Just a moment. Sorry. No questions. No questions. Does anyone has any questions? Francis, maybe I can change it to Francis.
If anyone has any questions, we can unmute you so that you can actually ask your question yourself. So we have a question here from Timo Kurtz. So okay. I've unmuted Timo Kurtz. So you can ask your question now.
Yes, can you hear me?
Yes, we can.
I can hear you.
Oh, wonderful. Okay. So I'm wondering if the signal from the germ-line stem cell, is that actually, does that need to be maintained throughout the lifespan of the worm to maintain repression of the chaperone? So in other words, if you were to deplete this cell, let's say with a laser afterwards, would you reestablish the proteostasis network?
Timo, that's a wonderful question. So we don't know the answer to that, and these are the kinds of experiments that we're actively investigating now. Is how many signals are there? So of course we've done genetic screens already to identify components, but I think you bring up what is a very important one, whether it's an initiator signal or a sustained signal. Since we know that one of the key targets is the transcriptional upregulation of the Jumanji 3.1 demethylase, it does give us an inside hint that we're looking for a signal that activates a transcription factor. And whether because the repression is tied into H3K27, we're thinking that you need to have a persistent signal. Right, because you can imagine that since you're going to need constant expression of the demethylase to prevent the H3K27 repressive marks. As soon as that imbalance is altered and we get a decline, then the H3K27 may then just competitively overwhelm the system. We don't understand the nature of selectivity at this point as well. How does H3K27 know which chromatin loci to modify?
Right. So have you tried to deplete the cells with a laser?
Yes, we are attempting that as we speak. It's a key experiment to do.
Okay, thank you.
Yes, thank you for the question.
Thank you very much, Timo. There's another question here from Francis Yen. So I've unmuted you, Francis. You can ask a question.
Oh, yes. Hi, can you hear me?
Yes.
I can.
Yes, okay. Thank you for the talk. It was very interesting. This is not my field at all. I was just curious if you think that intervention by nutrition is possible. Are these genes, can be influenced, these different chaperones, can the expression be influenced by dietary factors?
Absolutely. And I would just direct you to heat shock factor itself is directly regulated by SIRT1, which is, of course, directly regulated by NAD.
Right.
And so if there's no NAD or reduced levels of NAD, HSF cannot bind to the promoters to regulate the expression of all of its downstream targets. So I think it offers a very satisfying molecular explanation consistent why both sleep and caloric restriction are actually proteotoxic preventive strategies.
Okay. All right. Thank you very much. Thank you.
You're welcome. Thank you.
Are there any more questions for anybody? I think as we're running out of time, maybe we should draw a close to this. And if anybody has any further questions, then do feel free.
There's another question over here.
Oh, I see.
Can you see it?
Hwanai Kai. I'm sorry, I can't pronounce that name very well.
Yeah.
I'm going to unmute you so you can ask your question. Hwanai Han Kai.
Go ahead.
We can actually read it.
Okay, I actually can't see it anymore.
We can read it. As a critical window for HSR regulation is around 1 day to 2 from signals sent by the germ-line stem cell.
Yes.
So is it because at that moment germ-line stem cell starts to differentiate and produce oocyte?
So I guess I'm not sure what was the question.
The question was whether HSR, since HSR regulation is at the first or second day from the signal sent by the germ-line stem cell, right. He is asking whether it's because of the moment germ-line stem cell starts to differentiate and produce oocyte.
Actually, I don't think we know. We think it actually is uncoupled from that.
Okay.
Yeah, we don't know more.
And there's another question from Alexandra Stolz. Francis, can you unmute her, or shall I ask?
So Alexandra Stolz is unmuted.
Yeah, hi. I just wanted to ask whether there are certain transcription factors known that are activated upon kind of the maturation of the worm that might have a role or might play a role that you can speculate on. That this might have then also an influence on the kind of the production and transcription of the chaperones.
Yeah, that's a great question. Again, this is exactly the kinds of experiments we're doing right now. Since we know the genetic network that's initiated, both, well, repressed in one case, or initiated to provide protection between day 1 and day 2. And
but
so,
do you have any guess?
Oh yeah. I mean, so there's a set of proteostasis transcription factors. And I actually commented on them very early, and it's in the original cartoon on proteostasis. We'll call it, we got lucky and guessed correctly at the beginning. DAF16, HSF1, FA4, SKIN1, looks like it's tying into what is a stress network that is essential at that precise moment.
Okay. Do you have by chance any idea if this has also an influence on the autophagy genes and proteins?
Oh, absolutely. But that's what's fun about this, is that this is not just going to be about traditional heat shock stress or just folding and chaperones. This ties in beautifully into autophagy and into the proteasome as well. Absolutely.
Okay, thanks a lot.
Okay, we have one last question from Timo Kurz again.
Okay, Timo?
Yes, I was just wondering, do the CLIP1 and CLIP4 mutants live longer? Because that's what you would expect, no?
Yeah, they do.
They do?
Yeah,
okay.
Yeah, absolutely. Yes.
Okay, wonderful. Thanks.
Thank you.
Okay, I think we can thank Rick Morimoto for the beautiful talk, and thank you all for listening. And we hope to see you again at the next proteostasis webinar of Alfred Goldberg at February 4th, 2016. Thank you. Thank you, Rick.